Hi everyone, I'm Denise Colombano. Today I'm going to be talking about strength in numbers and how I think it can be used to advance estuarine ecology through data synthesis and collaboration. I'm a Delta Science postdoctoral fellow at UC Berkeley, where I work with Professors Stephanie Carlson and Albert Rui as part of a newly formed estuarine ecology team. We work on several Delta projects, ranging from food webs to fish community dynamics, and our main focus is trying to understand how climate change may impact the San Francisco Bay Delta. To give you some more background on me personally, I'm an avid field ecologist, someone who loves natural history and fish, especially in estuaries and tile marshes. I'm an enthusiastic data scientist. I love learning new ways to code and visualize data. And I'm also a basketball superfan. So I watch at least 82 Golden State Warriors games each season, and I say at least because we have been very fortunate to go to the playoffs in many of recent years. There are two pillars of my academic work. The first is ecological synthesis, leveraging multiple data sets in new ways to answer conservation and management questions. The second is team culture, participating in collaborative research teams that are hardworking and dedicated, but also unselfish and fun. In terms of ecological synthesis, I focus mainly on these elements. How does habitat complexity, environmental fluctuations, and habitat connectivity influence ecological responses in the estuary, and particularly for fish communities and food web dynamics? So the purpose of the ecological synthesis that I do is to generate knowledge and insight into the ecology of our highly dynamic and ever-changing system. In terms of team culture, I've come to the realization over the last 10 years that my ideal research team has a culture of hard work and dedication, as I mentioned, but also respect, trust, unselfishness, creativity, and joy. So basically what I'm saying is that my ideal research team is the Golden State Warriors. And let me show you what I mean. I want you to watch this team's chemistry and how much they share the ball.
Third longest streak in NBA history with a three made. Draymond on the kick out. Better pass. The flyby, return it. Wide open, good!
NBA's MVP last season. He averaged just under 24 a game. Golden State Warriors last season, 39-2 here at home. Oh,
Since I'm just one of many scientists who adore the Golden State Warriors, I decided to interview others to get their insight on team culture. What does the team motto mean to you?
It means that everybody has a responsibility for the shared success of the team.
There's more than each person's individual contribution to the team. It's all about the team first mentality.
I think it's definitely recognizing everybody's strengths as well as their weaknesses to be able to play up what one needs at the time.
It's very much a team attitude, right? It's not like strength in our star player. It's strength in everybody knowing their role and, falling into that team dynamic.
That everyone's able to contribute and that the team is giving back to everyone equally.
How does this apply to research teams?
Being able to essentially decenter ourselves and work on projects together in unison, but always being able to help and collaborate with others allows us to do more. It allows us to kind of stretch the boundaries of our own disciplinary silos and be able to work on projects that maybe is not our first expertise or would be our first choice, but allows us to really stretch that muscle of a brain so that way we can better understand how the entire system is connected.
Many of you in the IEP community may remember that I started out working on the Delta Largemouth Bass Project, led by Doctors Andy C. and Louise Conrad at UC Davis. And this was my first experience with a collaborative research team that had strong leadership and a culture of hard work and dedication, but also that unselfishness and that joy. So as a technician, I learned technician things. I learned to drive a boat, sample vegetation, and identify delta fishes. Here are some of the most common species in the interior delta fish community, a majority of which are non-native. We found that largemouth bass are associated with warm water and submerged aquatic vegetation, signaling a self-sustaining littoral community dominated by slow-moving warm water and invasive species. And our team published numerous peer-reviewed publications, a curated database with a full metadata report, and me, a technician, I was invited to think critically about what I was observing, ask challenging questions, struggle to learn new things like data analysis, and even help write these peer-reviewed papers. And as a result, I became a published author, which not only helped my CV, but also gave me insight on the entirety of the publication process. Being one of many contributors to the Delta Largemouth Bass Project, with all of our team members and all of our skill sets and all of our data sets, was a very formative experience for me and prompted me to pursue graduate school. In addition to this amazing experience with a healthy team culture and wonderful mentoring, my interest research peaked when in 2011 we started sampling fishes of the North Delta, where instead of largemouth bass and red ear sunfish in every single survey, we were capturing these absolutely beautiful native fishes: white sturgeon, delta smelt, Chinook salmon, hitch, splittail, and blackfish. Not only was I in fish biologist heaven, but this project sparked my academic curiosity in the question: what makes certain areas or habitats in the Delta good native fish habitat? With lots of guidance and coaching from my mentors and my peers, I enrolled in graduate school at UC Davis, where I joined Dr. Peter Moyle's lab in the Sassoon Marsh Fish Study, truly the honor of a lifetime. This is a long-term monitoring study that's been carried out by dozens of graduate students, hundreds of undergraduates and volunteers. It's a heroic team effort, and I expect that many of you in the crowd have also been on these boats, pulled these nets, admired these native fishes such as tule perch, starry flounder, and Sacramento sucker, and learned a ton about the ecology of the estuary, just as I have. Now, that being said, I really struggled at times during graduate school. I was in charge of my own research projects for the first time. I was spending 50 to 60 days a year sampling the marsh while taking classes and TAing, and I was helping with other field projects too. But that all changed once I received the Delta Science Fellowship. It gave me the freedom to think deeply about my questions and to learn new skills like coding and modeling that helped me answer my questions. So I want to express my gratitude for the opportunity to participate in the Delta Science Fellows Program, which has truly been a game changer for my career training and development. For my dissertation work, one of the things that really interested me most about Sassoon Marsh is that not only does it look very different from the interior delta, but it's long been considered an important fish nursery in the estuary, for the entire estuary, for numerous species, for native and non-native species. So I decided to look more into the roles of specific habitat types as nurseries. In the nursery role hypothesis, the idea is that tidal marshes often function as nursery habitats by providing vegetated cover and food resources, which together enhance growth and survival of juvenile fishes and ultimately facilitate migration or recruitment to adult populations. Now, Sassoon Marsh has experienced extensive landscape change due to diking and draining of tidal marshes. For instance, this is a beautiful aerial photo of Rush Ranch taken by Dr. Carson Jeffries from a helicopter in 2017, which was a year of record high flows. So notice that there's a reticulate tidal marsh channel network, a gradual upland transition zone, and this is surrounded by managed ponds that at the time were overtopping. There are large slough corridors connecting the whole marsh to the open water Sassoon Bay. And despite the fact that it's been highly altered, it's still largely managed to produce food in ponds that are managed for waterfowl. This is in stark contrast to much of the delta, which has not only been extensively diked and drained, but a majority of its marsh and floodplain habitats have been replaced by agricultural fields. And as a result, there's much less net primary productivity from the system, as shown by Klern et al. In their recent paper. I teamed up with Dr. Amber Manfrey, a postdoc in the Moyle Lab at the time. I'm sure many of you know her and her beautiful maps. Together, we quantified the area of a given habitat type, whether it was managed pond, tidal marsh, upland, or a combination of these three that we just called land, within 200-meter boundaries of the channel margins. And we divided those areas by the area of open water to quantify land to water ratios. To orient you, this is a two-dimensional ordination. It's a dissimilarity matrix of samples, where the gray points represent study sites, the arrows represent significant fitted environmental variables, and the ellipses represent two categories: sites with a higher land to open water ratio versus sites with lower land to open water ratio. And in ordination space, things that are closer together are more similar. What we found for each species that we examined is that while they may have started out in one habitat as juveniles, they typically shifted to other habitats as they grew to adulthood. For example, striped bass moved from shallow, warm, vegetated habitats as young of the year to cooler ponds as adults. Splittail had a similar pattern as striped bass. In contrast, tule perch have smaller home ranges than striped bass and splittail, and they shifted from marsh habitats to marsh habitats next to ponds. Starry flounder, on the other hand, were strongly associated with deep open water dominated sloughs, which was quite different than the other species. The key takeaways of this study were that yes, individual habitat types functioned as nurseries for fishes, but overall, the habitat mosaic is what supports multiple life stages of fishes with diverse life history strategies. Put another way, a habitat mosaic has diverse opportunities for fish to move around and to find favorable conditions for foraging and growth. And this is a synthesis effort that was only made possible due to our strength in numbers. Numbers from an amazing long-term monitoring data set, but also a diverse skill set among the researchers on the team. After looking into the nursery role hypothesis, my next goal was to look at the trophic relay hypothesis to better understand the role of tidal marshes in providing food to fishes in the estuary. So here's a conceptual model of a trophic relay, by which intertidal marsh production is transferred to the open estuary through a series of predator-prey interactions. And so the idea here is that primary consumers feed on primary producers in the marsh, then secondary consumers feed on primary consumers, and ultimately this relays marsh production to higher trophic levels, which then move out into deeper habitats. It's based on the idea that there are shifting interaction zones between predator-prey as a function of habitat and tide. So here's a tidal marsh during an incoming high tide, where the red lines indicate hypothetical hotspots of predation on marsh nekton. And this would be up against the marsh surface edges, where small fish attempt to seek refuge in deep water. Here's the same tidal marsh during outgoing low tide, where the red lines once again indicate hypothetical hotspots of predation. But now they're in the intertidal subtidal confluences, the subtidal open water confluences, and the subtidal open water areas. We tested this hypothesis with three main approaches. Our first was to use Thomas Hanley, a master's student at the time, his amazing bathymetry data set, showing this transition from low elevation marsh and subtidal channel beds to high elevation marsh with intertidal channel beds. We also used gill nets that were randomly stratified by habitat type along this gradient and by tide, whether the tide was incoming or outgoing. Here are photos of some of our awesome team members, including Emma Davidson, an intern at the time who now works on the CDFW tidal wetland monitoring team, and was clearly excited about the striped bass that we caught in an intertidal channel during an early morning high tide in the middle of summer. We looked at the diets according to each species feeding guild. So for example, planktivores, benthivores, or piscivores. We use spatially explicit generalized additive mix models and a soap film smoother to look at fish abundance. So here, small-sized planktivores were overall more abundant at lower tides, but particularly during incoming tides. They were less abundant at higher tides when there was deeper water. In contrast, piscivores had the opposite pattern. They were less abundant at lower tides in shallow water and more abundant in high tides, but particularly in deeper pockets of water in the subtidal open water habitats and confluences. So this started to make some sense in light of the shifting interaction zone hypothesis. Small fish can seek refuge in shallow water to avoid predators. Surprisingly, we found that very few fish had empty guts, and in fact, there were high gut fullness scores across species and guilds. Planktivore diets were dominated by mysids, larval fish, detritus, and some amphipods. Benthivore diets were dominated by amphipods, isopods, nematodes, clams, and detritus. Piscivore diets were all over the place. Larger striped bass ate anything and everything, including small fish, crayfish, shrimp, crabs, mysids, amphipods, and even chironomids. And Sacramento pike minnow, which has been shown to have very similar diets to striped bass in the Sacramento River, only had detritus in their guts, which we think might be an artifact of their propensity to evacuate their guts when they're captured in gill nets. Our top-ranked gut fullness model also yielded some really interesting results. Regardless of species, during incoming tides, gut fullness was highest in subtidal reaches. During outgoing tides, the pattern was slightly opposite, where gut fullness was high across the entire elevation gradient and including in these shallow intertidal reaches. Together, we inferred that small fish actively move into marshes to feed when they become inundated, and then predators move into the subtidal areas when they become sufficiently deep to ambush their prey. Overall, after the marsh has been flooded and the tide is going back out, fish bellies are full across the board, regardless of species or feeding guild. Based on our findings, we developed a simplified conceptual model where we hypothesized that tidal marsh hydrogeomorphology can drive tidal exchange with emergent vegetation, which supports benthic invertebrates and benthivorous fishes, representing a benthic food web pathway. Alternatively, it can drive mixed resonance time and phytoplankton production in the open water column, which supports zooplankton and planktivorous fishes, representing the pelagic food web pathway. Now, overall, we assert that tidal marshes can support diverse food webs and foraging by multiple feeding guilds, including piscivorous fishes that ultimately can relay this production to open waters. Once again, this was an amazing strength in numbers exercise. But our strength in numbers is not just from our study alone. There is accumulating evidence from all sorts of other studies showing the ecological importance of tidal marshes to food webs and fishes. This is ranging from Sassoon Bay to South Bay and the North Delta. And here's a list of recently published papers that I encourage you to check out. So while this is amazing news, we are unfortunately already committed to a certain level of global warming. Whether it's 1.5 or 2 degrees Celsius, we are already committed to a certain level of sea level rise. And this means that our current and future restored marshes are vulnerable to going underwater. And Sassoon Marsh, the areas that are going to be the most resilient are those that are adjacent to uplands, whereas the managed ponds will be the least resilient due to subsidence and overtopping of levees. And I just want to give a quick shout out to the amazing people out there that are working on climate change adaptation strategies, like SFEI and the Delta Stewardship Council. I highly recommend checking out these online resources. Unfortunately, this isn't just a regional issue. This is a global issue. So for this paper, I collaborated with a team of salt marsh ecologists from around the world to synthesize information on climate change and tidal marsh food webs. And we found that among many stressors, sea level rise alone is going to cause channel expansion and conversion to open water, runaway pond expansion, issues with marshes that are trying to migrate but being constrained by existing infrastructure, but also opportunities to let marshes migrate into upland transition zones, which is viewed as the most promising strategy for marsh resilience under climate change. This work was one of many products resulting from a surf workshop in Alabama, which was another highly successful collaborative research team that I'm very fortunate to have been a part of. We revisited this textbook, Concepts and Controversies in Tidal Marsh Ecology, which was published in 2000. It was a very neat experience for me because I was able to meet many of the original authors, Mike Weinstein, Jean Turner, Carolyn Curran, Linda Deegan, Ken Abel, Lawrence Rosas, Tom Manello, Juice Sebrian, Rod Connolly, and more. And the main conclusion of this workshop, after days of presentations and discussions, is that we don't have the luxury to sit around and argue about small details in textbooks anymore. We need to act now to prevent widespread loss of marshes, their food webs, and their support of fisheries. And the workshop coordinators made it clear to us that those of us in the younger generations, we are being handed the torch now. It's critical that we get involved and become leaders and thinkers and problem solvers to work toward marsh preservation. So this experience motivated me to write another Delta Science Fellowship proposal, this time on climate change impacts on the Delta, and also to join the IEP Climate Change Project Work Team, which I encourage everyone to check out if you haven't already. So as I said at the beginning of this talk, for the last 2 years, I've been working as a Delta Science postdoctoral fellow at UC Berkeley, looking at climate change impacts on fishes, which I will be presenting on in the next session. So this is an absolutely wonderful team with great, thoughtful leadership. And I have now stepped into a new role of mentoring, which I view more as an assistant coach position and something that I'm extremely excited for. We just started working on a CDFW Prop 1 funded project on climate-driven food web mismatches. We hypothesized that climate change may shift phenological responses among food web components, such as phytoplankton, zooplankton, or forage fishes. And based on the direction and magnitude of change in timing and overlap among these components, there could either be a trophic match, leading to high juvenile recruitment of forage fishes, or there could be a trophic mismatch, leading to low juvenile recruitment of forage fishes. Not only is this project another strength in numbers team effort, but we are hoping to leverage the strength in our data sets by using an open science framework. So our goal for this project is that every step is documented in a seamless, reproducible workflow, starting from the beginning and going all the way to the publication of the report. We also hope that this workflow uses open source software, open data, and publishes the report open access for full transparency, sharing, archival, and discoverability. But this is not just a collaborative synthesis team at UC Berkeley. This is an effort that's advised by Jim Hobbs, Noah Knowles, Lisa Lucas, and Jim Klern that leverages numerous publicly available data sets and builds on much of the amazing work that has already been done in the IEP community. Now, back to more analogies. The Golden State Warriors team culture, one that is collaborative and based on trust and unselfishness, can be applied to individual research teams, of course. But it can also be applied to our IEP community at large. We all play different roles and we all have different strengths and weaknesses. Some of us contribute to model development and application, new tools and approaches, advancing DEI, extension and public engagement, data curation and creation, knowledge generation, synthesis, and linking disciplines. Collectively, our contributions are rivers that flow into the same estuary of products and outcomes, one that advances our ability to manage a healthy ecosystem despite its many challenges. So with that, I want to thank everyone in the crowd, whether you are pulling nets on a boat, entering data, publishing data, writing reports, granting permits, balancing budgets. Whatever it is, thank you for your contribution. I appreciate you. And as a long-term data synthesizer now myself, I couldn't do my job without you. Now I'm going to leave you with a number of different ideas to reflect on, whether you're a mentor or a mentee, on how to cultivate a successful, healthy team culture.
My advice would be to kind of personalize the mentoring style and custom fit the mentoring style to fit each mentee. I would also encourage them to create a culture, kind of a larger culture for everybody to buy into. It creates a standard and it helps everybody hold each other accountable. And if you can get everybody to buy into that culture, then it kind of almost becomes like its own self-sustaining system. And within that system, you celebrate you celebrate heavily each other's victories, even small ones, and you lift each other up when people kind of mess up and things like that.
If you're studying the ocean, you could think sort of like competitively that you're trying to, I don't know if win is the right way to think about it, but you're definitely trying to like accomplish getting this knowledge. And thinking about that in the team way versus, trying to compete with other individuals, especially if they're on your own team, I think is a healthy way to look at it.
Getting everyone involved with each other's work in as many ways as possible is going to create a space where you're all excited to work with each other, that you see your efforts as being like communal, as well as being extremely individually beneficial, because suddenly writing that paper or finishing that field work or getting that experiment done is no longer so onerous on you by itself. It's a team lift.
Your advisor is not going to be that one coach who knows everything. That's why you have your committee. And that's like analogous, I guess, to that coaching staff. Your committee is that coaching staff.
Oh, good point.
You have everybody on your committee. Like on my committee, I have somebody for ecology, someone for like cell biology, somebody for physiology. That's my coaching staff. And then head coach is my advisor.
You need to have plasticity in order to understand how you can interact with and help out students that frankly are going to be different from each other, right? So the students that I have that are in the lab right now, I use certain strategies for some of them and certain strategies for others. I don't use the same strategy for everybody because not everybody is coming in with the same knowledge about their system, with the same interests. There are different people just generally based off of where they grew up, what their family structure was like. So for me, it's all about learning from their perspectives and then thinking about how can we co-produce the ways in which we scaffold your learning, knowing full well that for me, I'm taking myself out of it and really just being a listening board.
With that, I'd like to say thank you to all my funders, thank you to all my collaborators, thank you to the Carlson and Ruhi Lab and the Moyle Lab and the C Lab for everything that you've taught me. And please stay tuned for Lauren Damon, who's going to come up next with a wonderful plenary.